हेलो दोस्तों, ये एक ऐसा वीडियो है जो आप सबको देखना चाहिए क्योंकि इसमें कुछ बहुत ही इंटरेस्टिंग चीज़ें हैं जो हमारी लाइफ से जुड़ी हैं और जो एक मेडिकल पर्सन की लाइफ में उसको पता होनी चाहिए। सो आज का जो टॉपिक है oogenesis एंड फर्टिलाइजेशन। दिस टॉपिक इज़ वेरी क्रूशियल इन वंस अंडरस्टैंडिंग ऑफ अ ह्यूमन लाइफ, इट्स बिगिनिंग एंड हाउ there are stages in the development of ovary, oogonia, and the germ cells in the females, the ovum. So let us start with today's session. We are going to deal with oogenesis today, and oogenesis involves various things. Where they are located, the oogonia, the primary germ cells. Some people write primordial germ cells. So where they are located in the ovary. Before the ovary develops, they are primarily, resident of yolk sac from where they come. And as you know, by this time you must be knowing that everything in the human body is coming from the inner cell mass or the epiblast cell. So now the recent books are mentioning that even the oogonia or spermatogonia or the primary primordial germ cells, they are derived from the epiblast via yolk sac, and the wall of the yolk sac by the amoeboid movement they will come into the places where the genitalias or the gonads, rather the gonads have to be developed later on. So they are going to be residing there. In case of female, they'll be sitting in the ovary. In case of males, they'll be sitting in the testis, and the testis comes out for the various reasons in the males, but the ovary remains in the pelvis. तो टेस्टिस तो बाहर आता है मेल में, बट ओवरी अंदर रहता है, एंड इनसाइड द ओवरी यू हैव द oogonia. देखो, ओवरी और टेस्टिस की जर्नी अलग है और उनमें जो गैमेट्स डिसाइड करते हैं इन प्राइमरी फॉर्म, उनकी भी जर्नी अलग है. इन द मेल्स द जर्नी स्टार्ट्स, द डेवलपमेंटल जर्नी एक्चुअली स्टार्ट्स आफ्टर द puberty or at the puberty, and in case of female it starts before the birth. So by the time of 5th month of female fetus, they have all the oogonia formed, and mitotically they have progressed into a stage called as primary oocyte, and in that primary oocyte even they start to divide, and suddenly they stop at a stage called as prophase 1, where the chromosomes are closer to each other in the dictyostene, dictyotin or dictyolotine, this phase of prophase 1, dictyotin phase or diplotine phase we call it. That phase is where the chromosomes are about to separate, but at that end stage of that prophase dictyotin phase, they remain with each other till the puberty, when the hormonal things take over and they'll again separate to complete that meiosis 1. So the beginning of oogenesis is before the birth, whereas spermatogenesis, the development will start actually after the birth. Technically speaking, all the germ cells, they will start to develop before the birth only, but their transformation into the gamete starts after the birth in case of male, and in the female they are already started before the birth. So this is a one major difference, where you have oocytes arrested in females at the birth, where there are no spermatocyte, there are only spermatogonia till the puberty in case of males. So keep this in mind. And obviously the site of oogenesis is ovary, and only the last stage is completed in the fallopian tube, that we will see as you move forward. The time of oogenesis, obviously it is different at different stages, and it's a distinct feature than spermatogenesis, as I have explained. Only one mature ovum is formed by one oogonium. This is another a must know point. One oogonia gives rise to one ovum, and one spermatogonia gives rise to four spermatids and spermatozoa, right? So spermatozoa, four, are derived from one spermatogonium, then one oogonium, only one. Why it happens and why there is a need for this, again I will explain as we progress. Oogenesis doesn't complete without fertilization. Okay, the process, the end stage requires male chromosome to be there, whether it's a Y, whether it's a X, no problem, but it has to enter through the ovum, and then only you will have oogenesis completing. If it doesn't complete, it remains incomplete and ovum is expelled from the female body through the menstruation in arrested phase only. Which is that phase, why it happens, again we are going to deal with it as we go forward. दोस्तों, ये जो ओवरी है, दिस इज अ स्कीमेटिक ऑफ ओवरी, जिसमें आपको दिख रहा है कि ओवरी इज लाइंड बाय वन इपिथेलियम जो रूडिमेंटरी होता है. अब मैंने सब जगह ये नहीं बनाया, बट आपको समझना है कि ऐसा वाला इपिथेलियम सब जगह होता है जो एक क्यूबॉइडल लाइनिंग होता है. अब ये क्यूबॉइडल लाइनिंग पहले लोग मानते थे कि इसी से ये सब oocytes डेवलप होती है, इसीलिए इसको जर्मिनल लेयर भी बोलते थे. बट अब ये प्रूव हो चुका है कि ये मिसनोमर है. oogonia पहले से ही ओवरी के अंदर होती है, और जैसे जैसे मेंस्ट्रुएशन हर महीने होता है, एक एक दो दो oogonia डेवलप होके फॉलिकल में दे विल कम आउट एज ओवम थ्रू द ओवुलेशन. तो ये तो जान लो कि जर्मिनल लेयर जो होता है मिसनोमर है. इट्स अ क्यूबॉइडल लाइनिंग ऑफ पेरिटोनियल कवरिंग, जो एक मेसोथेलियल लेयर होता है, एंड दिस इज व्हाट द लाइनिंग ऑफ ओवरी इज एक्चुअली. अब ओवरी में होता है एक कॉर्टेक्स जिसमें सारे फॉलिकल डेवलपिंग स्टेज में होते हैं. उसमें एक मेडुला होता है जिसमें ब्लड वेसल होती है, कनेक्टिव टिश्यू होती है जनरली. और आपको दिख रहे हैं कि कोर्टेक्स इज द मेजर एक्टिविटी सेंटर ऑफ ओवरी. तो कोर्टेक्स में क्या है, डिफरेंट ग्रोथ फॉलिकल्स, ग्रोइंग फॉलिकल्स, फॉलिकल्स विच कंटेन्स द oocytes, करेक्ट. अब oocyte जो होती है उसकी जो डेवलपमेंट होती है, इट इज बेस्ड ऑन द हार्मोन्स विच आर देयर coming into the pubertal life in the life of a female. If everything is correct, if everything is going as per the norms, this is what going to happen. There will be follicles, there will be ovum and ovulation. Okay. अब फॉलिकुलर सेल का काम होता है इसकी ग्रोथ को सपोर्ट करना. कुछ हार्मोन्स जो होते हैं, ईस्ट्रोजन एंड लेटर ऑन इवन प्रोजेस्टेरोन, आर secreted by these follicular cells. This follicular cell also gives rise to one of the membrane which protects the oocyte and the zygote later on, and this membrane is known as zona pellucida. What is this membrane is known as? Zona pellucida. The zona pellucida covers your oocyte. It's vitelline membrane inside and zona pellucida is on the outer tougher side. Okay, so zona pellucida is again derived from the secretions or the contributions from the follicular cell, and partially it is also contributed by the oocyte itself. So it's a combination of these two which will give rise to this tough layer surrounding the oocyte known as zona pellucida. The layers or the cells which are covering oocyte are follicular cell, also known as granulosa cell, and you have additionally the cells coming from the ovary stroma. This area is the stroma of ovary, and they are the connective tissue cells, they are the fibers, and few cells will differentiate into the theca interna and theca interna, two layers. One of that later on transform into the hormonal layer. Okay, it secretes again estrogen and progesterone. So theca cells will also contribute to the some of the hormonal function of later on this follicle. And you may have heard of this corpus luteum. So they are the part of that corpus luteum later on after the ovulation. If there is pregnancy, the luteum lasts for around 4 months, and then if it is doesn't, if there is no pregnancy, the luteum will be transformed into a white fibrous scar. We call it as corpus albicans. So these cells are important to notice. Oocyte residing in the shell of follicular cell, additionally cushioned by the theca cells, and this is what ovary contains. अब जो फॉलिकल होता है ओवरी का, उसके कुछ स्टेजेस होते हैं, जैसे प्राइमोर्डियल है, प्राइमरी है, सेकेंडरी है, टर्शियरी है, एंड टर्शियरी विल बिकम मैच्योर टू प्रोड्यूस द ग्राफियन फॉलिकल, और जिससे ओवम बाहर निकलेगा, ओसाइड बाहर निकलेगी, एंड द स्टेज विल बी कंप्लीट एंड इट विल बी एब्जॉर्ब इनटू द फैलोपियन ट्यूब नॉर्मली. ये जो फॉलिकल होते हैं क्यों इंपॉर्टेंट है? बिकॉज फॉलिकल के बगैर कोई हार्मोनल एक्टिविटी होगी नहीं, ओवुलेशन होगा नहीं. तो इसका जो पर्पस है ऑफ हैविंग द फीमेल गैमेट एंड इट्स डेवलपमेंट ऑल द वे, ये पर्पस ही सॉल्व नहीं होगा. तो फॉलिकुलर सेल भी बहुत ज़्यादा इंपॉर्टेंट है, जिसको हम ग्रेनुलोजा सेल भी बोलते हैं. तो ये जो हम देख रहे हैं, दिस इज अ प्राइमोर्डियल फॉलिकल with the squamous nature of follicular cells. Initially they are not well developed, so they remain flat with very less scanty cytoplasm, and you can see the nucleus of oocyte. Now this oocyte is the primary oocyte, and you can see the covering of this called as zona pellucida. Now again, the oocyte with the ooplasm, the zona pellucida, and the nature of this follicular cells changing under the microscope. If you observe this kind of follicle with the cuboidal or columnar nature with a single lining surrounding the oocyte, this will be called as primary follicle. ओके, अभी ये स्लाइड में आपको दिख रहा है प्राइमोर्डियल फॉलिकल से कैसे प्राइमरी फॉलिकल बनाया और कैसे ये प्राइमरी फॉलिकल सेकेंडरी में ट्रांसफॉर्म हुआ जैसे ही ये मल्टी लेयर्ड फॉलिकुलर सेल बन गई. So this cell will accumulate over each other, and they are coming through the stroma of the ovary. Okay, and this is your secondary follicle. So follicle का growth follicle stimulating hormone जो आपका ovary जो pituitary से आता है into the ovary blood stream, and this will stimulate the growth of the follicle in the puberty. Now you see सेकेंडरी फॉलिकल और प्राइमरी फॉलिकल में सिर्फ लेयर का फर्क है, एंड द साइज ऑफ oocyte आल्सो इंक्रीजेस लिटिल बिट. अब ये हो गया मैच्योर फॉलिकल, जिसमें आप देख रहे हो कि ये जो सेल्स है कितनी ज़्यादा बढ़ गई है. दिस आर ऑल योर फॉलिकुलर सेल्स. दिस इज द oocyte, दिस इज द इनर मेम्ब्रेन, वेरी थिन मेम्ब्रेन ऑफ वी कॉल ooplasm covering the ooplasm. This is your vitelline membrane. Okay, so the membrane is vitelline membrane, and this is the oocyte ka ooplasm or the cytoplasm, and outer to that you have a amorphous nature membrane, a little bit of tougher membrane called as zona pellucida. This is a very important membrane because इसके बगैर फर्टिलाइजेशन होगा नहीं. दिस इज अ पर्टिकुलर रिसेप्टर कॉल्ड एज zona pellucida receptor, ZP receptor. Okay, ZP 1, 2, 3, 3 nature are there. Third number is very important because ZP3 is very specific for the attachment of the acrosomal head of the spermatozoa, and this is the, we can say, the initiator of the fertilization. These receptors bring, and with that chemotactic nature of the ovum, few substances secreted by it inside the fallopian tube, the sperm will swim into the uterus and uterine tube and reach into the ampulla part of the fallopian tube to fertilize this ovum. And you can see the ovum surrounded by lot of follicular cells. Inside that you have the development of a cavity. We call generally to this cavity as the antrum. In the medical language, any antral thing is having a cavity. So antrum is the cavity inside the follicular cells of this follicle, or the graphene follicle rather, and The intimate cells which are covering your the surface of the oocyte, here we call it as corona radiata cells. They are nothing but the follicular cell immediately adhered to the surface of this oocyte. These cells need to be penetrated by the incoming spermatozoa, and then only the fertilization is possible. Okay. There are terminologies like cumulus, and this cumulus is again nothing but the follicular cells, okay, surrounding the follicle. And cumulus oophoricus is this cells, okay, which are over which they are cushioning your oocyte. So there are few things you need to be aware before you proceed into the next topic. अभी आगे चल के क्या होने वाला है आपको पता होना चाहिए. Fertilization, where the union of male and female gametes leads to the formation of zygote in a sequential manner, and that sequence start with the penetration of sperm into the corona radiata, then it binds with the zona pellucida, then it ruptures the zona pellucida at one particular area through the enzymatic action, then it gets inside its head and tail gets inside, rest of the thing remains outside. There is no cytoplasm it brings with it. So only this nucleic material or the nucleus or the chromosomes of the male which are important to unite with this nucleus female to form the one complete diploid state of this ovum or the zygote which is formed after the union. This is the sole purpose of fertilization, is bringing your nucleus of secondary oocyte and sperm together, and again they form a diploid cell called as zygote. That is where the birth of an organism, human being, takes place during the process of fertilization, where the chromosome material of male and female unites after so many barriers, passage of these barriers. You have coronary data, you have zona pellucida, you have vitelline membrane, and then the nucleus will be residing here. It will become, obviously the two nucleus become similar in nature, and they for temporary period they remain like that, and they duplicate their material here, and then they will unite to form the zygote, and which immediately undergoes a lot of repeated cycle. We call it as cleavage, you know, before implantation. So this is the fertilization which is happening in the ampulla of fallopian tube. There's something that you need to know: acrosome reaction or zona reaction. What happens when the acrosomal head of the spermatozoa enters? There's a reaction happening all around in the oocyte. Okay, there's a calcium wave that runs. There is a change in the zona pellucida metabolic properties which will render your zona pellucida impenetrable or resistant to the penetration from another sperm, preventing polyspermy. So no sperm can enter after the first sperm enters into this ovum. The entry of all others are avoided through a reaction over the zona called as zona reaction, where it changes its metabolic property, rendering it non-penetrable to the other sperms. Okay, and this is the the beauty of fertilization, where the care is taken not to form too many, I would say, chromosomal combinations. Only one is entered. Polyspermia is not generally permitted by the nature only, and there are no chromosomal anomalies normally taking place. Yes, there it can happen in some simultaneous penetration of two sperms. So those zygotes will be non-viable. If there is a multiploidy, there is a polyploidy, the zygotes cannot survive. Only the normal zygote with the normal nature of chromosomes will be surviving there, and it will undergo the further process. So remember this: secondary oocyte, Okay, somewhere here in the perivitelline place you will have that polar bodies there, the sperm entering here uniting with the secondary oocyte, and later on this secondary oocyte will complete its final division. I will enlist the stages of oocyte and its development, but remember, after the entry of sperm during the fertilization, this secondary oocyte replicates, it divides, okay, to form another polar body which will go there with the 23x chromosome, or yes, 23x chromosome, and one pronucleus remains with the purely 23x. So this 23x and this 23x or y unite to form a zygote, whether male or female is decided here only, and then they are later on dividing like a normal cell, duplicating their chromosomal material, and they undergo the cleavage process. So this is how the further process is going on. So fertilization is important to maintain the normal chromosomal number in the cells, to form a new organism, to undergo variation, to maintain the species continuity. A lot of things depend on the fertilization, and obviously it is very, very important. Now this is actually the normal oogenesis process happening in the ovary and the end stage in the fallopian tube, that you have seen how it happens during the fertilization. The primary germ cells or primordial germ cells, we call it as oogonia, are there in the ovarian cortex. Before birth only, they will mitotically divide to produce the primary oocytes. Around 5 million of primary oocytes are present in the females, and they are 5 months old inside the uterus. At the birth, they remain around 5 lakhs, okay, or 4 lakhs. Some books have this 4 rule, 4 million, 4 lakhs, and at the adult life, they only have 5,000 around oocytes. Out of that, only 4 to 500 will be expelled in the normal female's life. So primary oocyte is the phase which will happen before the birth. Before the birth, they undergo another thing called as meiosis 1, but it doesn't complete till the ovulation is there. So this is the landmark. Okay, primary oocyte does not form the secondary oocyte unless there is a ovulation impeding. Just before the ovulation, it will undergo the meiosis 1 to give you the secondary oocyte, and the secondary oocyte is expelled through the ovulation. Okay, so if there is a ovulated ovum, there is a ovum. After the ovulation, it is under the secondary oocyte category, because just before the ovulation, this primary oocyte is transformed into the secondary oocyte by the expulsion of this first polar body. This contains 23 chromosomes. This contains 23. This has 46 chromosomes. This has 46 chromosomes. Okay, and now secondary oocyte by meiosis 2 will form the mature secondary oocyte during the fertilization, and immediately it will be forming the zygote with the sperm. Now see, even that stage, if it has to complete, it needs fertilization to happen. Without fertilization, there is no meiosis 2, where it is arrested in a particular phase called as metaphase of meiosis 2, or the metaphase 2 may this is arrested. Unless there is a sperm entering into it, it does not give this second polar body, you know. So they conserve their energy, they conserve their cytoplasm for the right things, and that is why these arrests are important in the life of the ovum. See, when before birth it assesses that you cannot undergo secondary oocyte phase, so it arrests in the meiosis 1, and just it analyzes the right hormones are there from the pituitary. That means we are at the right age to expel into the abdominal cavity and to be fertilized by the spermatozoa. the ovulation will happen. The meiosis 1 is happening just before the ovulation. So this is very random process. This is very dynamic process. And then you have the secondary oocyte which remain arrested in the metaphase 2 till there is fertilization. After the fertilization only the oogenesis completes with the extrusion of this secondary polar body, and immediately this will form the female pronucleus inside that. When there is a male pronucleus and they will form a zygote. Okay, so oogenesis technically completes after the fertilization and it starts before the birth. During third to 4 month oogonia are formed. They repeatedly undergo mitosis. They form the primary oocyte which remain arrested in the meiosis 1 diplotin phase. Okay, and this is very I would say a thing to notice, where in the meiosis 1 phase where they are arrested in the prophase 1, the chromosome are together like they are having a romance with each other, and even in this meiosis 2 the chromosomes remains with each other like romancing again. Okay, so metaphase 2. So this is very stark, but this is how they remain together. Okay, in here they are not separated where they are arrested. In here they are not separated, they remain arrested till there is a fertilization. So conditions are, if the conditions are met of ovulation, the right amount of hormones, then only oocyte will replicate, will complete the its journey primary to secondary. If there is ovulation, yes, it will complete, and majority of the oocytes will undergo this journey from primary to secondary. And only in the life of a female, one or two will go to this journey where fertilization is going to happen. Conditional. Okay, if there is a fertilization, this stage will come. The final stage will be completed only by one or two out of that 500 which are expelled there. So imagine 5 millions, 4 to 5 millions are formed, only one or two are going under complete oogenesis, that is fertilization. And this is the journey of a oocyte from its inception into the female, I would say, fetus, into the adult life of a woman. So stay tuned with our channel and let us, you know, subscribe and bring more of your friends so that we can go ahead with some interesting sessions ahead. Thank you.